Hi, in the previous class we have seen about the advantages and disadvantages of colon targeted drug delivery systems. Today we are going to see about the methods used for colon targeted drug delivery system. So now we discussed about the methodology used for colon targeted drug delivery system. The variety of methods available in the colon targeted drug delivery system. Based on this, now we discussed about the first one, formation of prodrugs. What is meant by prodrugs? Prodrugs is an inert drug that becomes active only after it is transformed or metabolized by the body. According to prodrug, two types is there. First one is azo bond conjugations. What is meant by azo bond conjugation? Example, sulfasalazine drugs is mainly used for the treatment of inflammatory bowel disease in the colon area. It is 5-amino salicylic acid. It is one type of prodrugs. Next, glucuronide conjugation. What is meant by glucuronide conjugations? Glucuronide and sulfate conjugation is the major mechanism for the inactivation and preparation for the clearance of a variety of drugs. So next we are going to see about the methods used for cyclodextrin conjugations. What is meant by cyclodextrin conjugation? The hydrophilic drugs and ionizable cyclodextrins can serve as potent drug carriers in the immediate release as well as delayed release formulation, while hydrophobic cyclodextrin. What is meant by hydrophobic cyclodextrin? That is the lipophilic drugs can retard the release rate of water. Moreover, the most desirable attribute for the drug carrier is its ability to deliver a drug to a targeted site like colon targeted drug delivery system. So next method we have to see about the dextran conjugations. What is meant by dextran conjugation? Dextran ester prodrugs of metronidazole, that is anti-amoebic drugs, have been prepared and characterized. The next, dextran ester prodrugs of dexamethasone and methylprednisolone was synthesized and proved the efficacy of the prodrug. What is by proved? That is the therapeutic effect of the prodrugs is delivering the drugs to the colon targeted drug delivery systems. Methylprednisolone and dexamethasone were covalently attached to the dextran by the use of a succinate linkage. The next method is amino acid conjugations. What is meant by amino acid conjugation? The contribution to the metabolism and toxicity of the xenobiotics of the carboxylic acids. So due to the hydrophilic nature of polar groups like NH2 and COOH. COOH is a acid, NH2 it is a amino radical forms that is present in the proteins and the basics units is called as amino acids. They reduce the membrane permeability of the amino acids and protein molecules. In this amino acid conjugation, the various prodrugs have been prepared by the conjugation of the drug molecules to these polar amino acids. The next, non-essential amino acids. Non-essential amino acids, the plenty of non-essential amino acids is there. So now discuss about such as tyrosine and glycine, methionine and glutamic acid were conjugated to the salicylic acid derivatives. So now we have discussed about the hydrogel methods used for colon targeted drug delivery system. What is meant by hydrogels? Hydrogels are highly absorbent material that contains more than 90% of the water. Hydrogels are network of a polymer chain like hydrophilic, sometimes is found as colloidal gel. Next, hydrogel can be used for site-specific delivery of peptide and protein drug molecules through the colon area. The hydrogels are composing of acidic components and enzymatically degradable azo-aromatic cross-linkage materials. The next method is coating with pH-dependent polymers. The pH in the terminal ileum and colon is higher than in any other region of the gastrointestinal tract, and thus dosage forms which disintegrate at high pH range can be targeted into the region. A level of pH is higher in the terminal ileum region than in the cecum area. Next method we have discussed about the timed released system. It is based on the concept of preventing the release of drugs, that is 3 to 5 hours after entering into the small intestine. In this approach, the drug release from the system after a predetermined lag time according to the transit time of the mouth to colon area. The next method is designing the formulation using the polysaccharides. The polysaccharides are long chain carbohydrates like starch, glycogen and cellulose, and polysaccharides are react with water and using amylase enzyme catalyst which produces constituent sugar. The dosage forms enjoy the shielding effect of polysaccharides in the upper part of the gastrointestinal tract, and drug is released in the colon area by swelling and biodegradable action of polysaccharides. The next method is redox sensitive polymer coating method. The analogs to azo bond cleavage by intestinal enzymes, novel polymers that hydrolysis non-enzymatically by enzymatically generated flavins are being developed for colon targeted drug delivery systems. A common colonic bacterium, Bacteroides fragilis, was used as a test organism, and the reduction of azo dyes like amaranth, orange 2 tartrazine and a model azo compound 4,4 dihydroxyazobenzene were studied. It was found that the azo compounds were reduced at different rates, and the rate of reduction could be correlated with the redox potential of the azo type of compounds. The next method is very novel method used for colon targeted drug delivery system, that is bioadhesive system. What is meant by bioadhesive? Bioadhesion is a process by which a dosage form remains in contact with a particular organs for an augmented period of time. This longer residence time of drug would have high local concentration or improved absorption characteristics in case of poorly absorbable drugs. This strategy can be applied for the formulation of colonic drug delivery system. The various polymers including polycarbopyls, polyurethanes and polyethylene oxide and polypropylene oxide copolymers have been investigated as materials for bioadhesive systems. Let's come to the end of the session. I hope you all understand about the methods used for colon targeted drug delivery system. In the upcoming lecture, we are going to see about the evaluation and conclusion of colon targeted drug delivery system. Thank you.